Myself Komal Duhan, and now we are discussing about the next structure of DNA. Means basically three type of DNA which exist: aDNA, bDNA, and zDNA. See, we are discussing their difference on the basis of their properties, different type of properties, and they assign the DNA on different basis: A, B, and D. What we are study about in our previous video lectures, that we study about the bDNA. bDNA is discovered by Watson and Crick. So now we're discussing on the basis of their property. First property, that is helix hardness. aDNA always right-handed, B always right-handed, only the zDNA that is left-handed. Second property, base pair patterns. A carry the 11, B carry 10.4, and Z carry 12. Next is rise per base pair along axis. See students, that are not that much easy. You think about it, we have to focus on that difference, because every time in competitive exams, either in NET or after post graduation, they ask question from that A, B, and Z DNA comparative. Like for example, if question is that the rise per base pair along axis in aDNA, they give the four option, then right one option is 0.23 nanometer, bDNA carry the 0.34 nanometer, Z carry 0.38 nanometer. Now the next property, that is pitch. aDNA carry 2.46 nanometer, bDNA carry 3.40 nanometer, and zDNA carry 4.50 nanometer. Next is diameter. A 2.55 nanometer, B 2.37 nanometer, and Z carry 1.84 nanometer. Next is about the major aspect, which every time asked in competitive exams. What I studied, that is conformation of glycosidic bond. aDNA anti, bDNA anti, and zDNA alternative anti and syn. So next property, that is denaturation and annealing of DNA. Firstly, DNA double standard can denature heated or treated with chemicals. AT rich denaturation first 2 hydrogen bond, GC reason denaturate last 3 hydrogen bond. DNA denaturation is a reversible process, as denature strand can reannihilate again if cooled. The process can be monitored using the hyperchromicity. So what is hyperchromicity? What does that hyperchromicity read? It is used to monitor the DNA denaturation and annealing. It is based on the fact that single standard DNA give high absorption reading than double standard at a wavelength of 260 nanometer always. Using melting profile, we can differentiate between single standard and double standard DNA. So now we discuss about the graph curve that represent the hyperchromicity and the melting profile. Hyperchromicity always behave at a nanometer 260 nanometer, and it always observe with the help of spectrophotometer. See in graph, you can see that absorbance on a x-axis and temperature on a y-axis, they can give a difference like DS and SS, single standard and double standard. Heating of single standard DNA, little rise of AB, and heating of double standard DNA, high rise of AB. Next, melting profile continue. Melting profile can be also used to give an idea about the type of base pair rich area, using the fact that AT rich region denaturate first, that is low melting point, GC rich region denaturate last, that means higher melting point. So higher melting point always denaturate last, and low melting point always denaturate first. So make sure student, and always denaturation and low melting point AT rich, and higher melting point and denaturation always the GC rich. As you shown in graph, that single standard and double standard, standard always at the saturation point and double standard at their peak. AT rich, GC rich, and denaturation lower melting point and higher melting point. Next topic, that is about the Chargaff rule. Chargaff rule, according to the name of scientist Argim Chargaff, they said that 50% of nucleotide were purine and 50% were pyrimidine. A always equal to T, G always equal to C. Amount of adenine equal to thymine and guanine equal to cytosine. So they indicate the DNA is symmetrical. So next, DNA X-ray differentiation pattern, that was discovered by the London and Franklin's work on X-ray differentiation. X-ray was the method to study the structure of molecule, that DNA has no difference. And the last topic, that is function of DNA का main important function that help in hereditary DNA consists 4 base A, G, C, T that are held in linear that are held in linear array by phosphodiester bond through the 3' and A' position of adjacent deoxyribonucleotide. DNA is organized into two strands by pairing A to D and G to C on a complementary strands. So 3 into 10 raised to power 9 base pair of DNA in human are organized into haploid complement of 223 chromosome, like 46-23 pair, that stands for the 46. DNA provide template for its own replication and maintenance of genotype. So last one, that is a quiz complementary sequence.